Well, thank you. It's a kind introduction and gratifying to see how much this field has grown since we all got started working in this area, and to see so many people who I've worked with me distributed around the world, seeing their faces again here today in person. This is my first in-person meeting in 2.5 years, so it's really quite wonderful. What I'd like to do is describe some of the work that we've done over the past 15 years or so in the organ chip area, where we have been able to recapitulate human biology, disease states, and therapeutic responses in vitro, and give you a feel. There's so much going on now, and you'll get this from this meeting, but give you a feel of our approach and why we're so excited about it. Now, I've been at Harvard for, 38 years. But for the last 13, I've been the founding director of the Wyss Institute for Biologically Inspired Engineering. And the basic concept behind the Institute is that engineering has transformed medicine over the past 50 years by taking engineering principles, trying to solve problems. When we started this envisioned it about 15 years ago, we wanted to flip that on its head. We really felt that we were at a point where we now have uncovered enough about how nature builds, controls, and manufactures, that we could actually leverage biological principles to develop new engineering innovations. And this is what we call biologically inspired engineering. And we were lucky to start with a very large philanthropic gift, but we were challenged with taking on really tough problems. But if we could solve them, it would have transformative impact. And one of the biggest problems that I could see personally at the time was that the drug development model is broken. I think all of you are here because you know it. It costs billions to take a drug from the bench to FDA approval. There are many reasons, but one of them is that animal studies are required. They take years to complete. They're expensive. The reproducibility is not always so great. But the real problem is that, and there are ethical issues, but the real problem is that more often than not, they don't predict what happens when they go to clinical trials in humans. And in some areas, this is like over 90% failure, but on average, it's about 70%. So there's been searches for ways to come up with developing drugs faster, cheaper, and that are more effective and safer. And so when we started the Institute, and that's where we started. But as we got involved in this area, we've learned that it's much broader challenge than that. For example, now many drugs are biologics. Something like 40% of drugs in the development pipeline are biologic, therapeutic monoclonal antibodies, RNA therapies, CRISPR RNAs coming into mode. And some of them are so specific for human, they don't even cross-react with other species. And so right now we rely on non-human primate models, which have enormous ethical concerns. We learned during COVID that they're actually in short supply as well. But there's really a desperate need for human models, even in this case. And then one thing that we haven't talked about beyond drug development is this One Health challenge. I think Dan mentioned this briefly, which is that we now very clear that animals are often the sources of disease vectors, and COVID with bats was a prime example of that. And the way it's handled now is, they cull livestock, which is a huge economic problem, ethical problem once again, and it's not even clear it's effective. And there's no way you're going to have, labs developing animal models for every species, but preclinical models, MPS, organs on chips, that's one way you could actually confront this challenge. So when we started the Institute, we had about 5 platform areas, and I led one myself that we called biomimetic microsystems, but really it was developing what we eventually called human organs on chips. And the idea was to use computer microchip manufacturing methods that in 1989, 90, I started to apply to cell biological challenges working with George Whitesides to leverage these techniques in computer microchip manufacturing that had control over features at the same nanometer to micrometer scale that cells and tissues live at, to use them to create microchip-like devices containing living human cells that reconstitute organ-level functions, not cellular tissue, but organ-level functions. And obviously, this was to accelerate drug development and replace animal testing, but it would also be useful, it was very clear, to advance personalized medicine. And our first success, which I do think helped launch the field, or at least get recognition for the field, was work that Dan Hudd did when he was a postdoc with me, that we called a human breathing lung on a chip. Now, the idea was never to engineer a whole organ. I actually worked on tissue engineering in the 80s, had a company that did that. This was a total different mind frame. This was, I want to build a living three-dimensional cross-section through a major functional unit of the organ. And in this case, we focused on the alveolus or air sac. And I think you all know the lung, has the alveolus is a pretty simple structure. If you go look at an electron microscopic level, there's air, there's a single layer of alveolar epithelium, a flexible porous basement membrane, and then a single layer of capillary endothelial cells, and then blood. But what this doesn't show is that it's an incredibly dynamic structure in that every time we breathe in and out, the alveolus stretches and then it relaxes, blood is flowing, air is flowing. And so if you were trying to distill down, like what are the simplest design principles to build an organ? The first thing is that an organ are two or more different tissue types that come together and new functions emerge. And usually one of them is a vascular tissue. The other is that in our case, we felt that the mechanical environment was absolutely critical. So we wanted to have this dynamic flow of perfusion through the capillary networks, air, and also cyclic breathing motions. So this next video shows you how this works. At the top right is the chip. It's the size of a computer memory stick, optically clear, made out of flexible rubber. If you cut it in cross section, there are 3 hollow channels, each less than 1 millimeter wide. The middle channel is cut into top and bottom by another thin, this is PDMS membrane with engineered pores. We coat that with matrix. Then we put human lung alveolar cells on the top, human lung capillary cells on the bottom. We just recreated the alveolar capillary interface. The trick is that we have, oops, I'm sorry. I lost that one. Let's see if that works again. I'll let it go through here. I went through quickly, obviously. The middle channel has a thin membrane that has pores that actually can allow cell movement. This is coated with extracellular matrix. We have human lung alveolar cells up top, human lung capillary cells at the bottom, create the interface. And then the trick is that we have cyclic suction applied to the side chambers. Everything's flexible. This is polydimethyl siloxane, silicon rubber, and it stretches and relaxes at the same rate and degree as when we breathe. We then put air over the top to create an air-liquid interface, and then we could flow medium with or without immune cells to the bottom. And if we have endothelial cells on all 4 sides, as we tend to do now, we could even flow whole human blood without anticoagulants for a period of hours. So if this were to work, it should mimic an organ-level response. So imagine you have a bacterial infection up top. What happens is that there's a tissue-tissue signaling response. Cytokines are secreted by the epithelium. They activate the endothelium. White blood cells that were just flowing by now stick, roll, diapedese, or migrate across and then engulf because the endothelium express adhesion receptors. And now I'll show you imaging in the device. What you see here are fresh white blood cells. I always like to say we know they're fresh. They were from Dan, huh? They're labeled fluorescently. You don't see the endothelium and you don't see the epithelium because it's on the other side of the screen. To begin with, it's quiescent. They just flow by. Now, if we put bacteria on the other side, you get that signaling response, and these are pulled out under flow, which is important because the initial adhesion step for an immune cell to endothelium is shear stress dependent. And now you can do any imaging you want in the device, so higher magnification, 1 white blood cell right there, it finds a space between 2 endothelial cells that are unlabeled, goes through the matrix-filled pore, goes out of focus to the other side. Now you're going to see it by phase contrast coming up through the epithelium, and now you're going to see the white blood cells in red because the bacteria are labeled with GFP in green and you see them being engulfed. So you just watch the entire human inflammatory response at high resolution in this little rubber chip. And I think this really got people's interest. Now, for those of you who are new to the field, I show some results from that first paper. We were actually not funded to build organ chips. We were funded on nanotoxicology. And people in the field in those days were realizing that pretty large nanoparticles were getting across tissue interfaces. It was not clear how this could work. And so the lung is a major site for this. So we developed the lung chip actually initially to look at that. And we used simulants of smog nanoparticulates that are in air. We had a fluorescent reporter for injury in terms of reactive oxygen species, oxygen-free radicals. And it was interesting that if we exposed the particles to the chip, if we had it with physiological breathing motions, we could see injury with ROS, and that actually recruited immune cells. But if we did the exact same experiment without breathing motions, just statically, we did not see this response. So if you did this in a trans well, you would not see this response. We also could quantify absorption. So you're seeing a graph at the left, but at the right are images of what's in the effluent of the vascular channel when we put the nanoparticles in the airspace. And we saw an 8 to 10-fold increase in absorption based on physiological breathing motions. Now, this wasn't mimicking physiology. This was never heard of before. This was a prediction. So we actually developed an ex vivo ventilation perfusion model in the mouse where we could separate breathing from blood flow, and we totally recapitulated that response. And this was a full article in Science. And this is really, I think, one of the first times that you could see that you could mimic organ level physiology and pathophysiology in a little engineered device. Now, we started to talk to pharmaceutical companies, and they thought this was great, but they were more interested in toxicity models and also particularly disease models. That's really where the animal models were limited for them at the most. And so we hit two birds with one stone by developing a model of pulmonary edema, fluid on the lungs, by perfusing the FDA-approved cancer drug interleukin-2 through the vascular channel, and it's normally infused in a patient, IV. And it's known in patients that the dose-limiting toxicity is pulmonary vascular leakage and pulmonary edema. At time zero, with air above the epithelium, this is what it looks like. When you perfuse the interleukin-2 over 2 to 4 days, you see a meniscus of fluid filling the upper space, and it completely fills it by day 4. This is the same time course and the same dose that is seen in patients. Once again, when we quantify this, we only see this with breathing motions. We do not see it under static conditions. And again, we confirmed that this is true in vivo as well. And this is important clinically because most patients with pulmonary edema are on a ventilator where you can actually tune the physiological breathing motions and ventilation pressures. Now, this finding that mechanical deformation was important for this toxicity got my interest because my entire career, and we're talking now starting like my first paper was like 78, has been that mechanical forces are as important as chemicals, genes, which is now known as mechanobiology. And we'd worked on mechanotransduction for years, and I had identified one of the earliest signaling response that we could see in endothelium when you stretched the extracellular matrix was forces going through integrins to an ion channel called TRPV4 in the focal adhesion in 5 milliseconds. And that induced calcium influx, and it was upstream from all the other integrin signaling responses. And I knew that GlaxoSmithKline was working on an inhibitor of this. And they did not fund our work, but they gave us the drug, and we completely inhibited pulmonary edema in this chip with that drug. They, in parallel, did studies on, I think, dogs and rabbits in a cardiogenic pulmonary edema model. This is failure due to back pressure when the heart fails. And they got the same inhibition response. And we had back-to-back papers in Science Translation Medicine. Now, I'll never forget, we had one reviewer who said, this paper should not be accepted because it's too simple, there are no immune cells. And the other reviewer said, this is amazing because this paper is like synthetic biology at the cell, tissue, and organ level. They just showed you don't need immune cells for pulmonary edema induction by the drug, and it got in. And we have seen this again and again and again. And this is something we don't talk about enough about in MPS and organs on chips, is the ability to control individual control parameters, one at a time, 2, 3, 4 cells, forces, factors, gradients, et cetera. And when you hear about the interest in mechanism of action of both drug efficacy and toxicity, this is when these systems provide enormous value. So this one model, this lung alveolus chip, has provided proof of principle for a disease model, a drug toxicity model. We've also published papers where we've shown toxicities with microfluidic organ chips that were never seen in animals, but were But for example, a lupus drug, a therapeutic monoclonal that no problem in animals, but there was pulmonary embolism in patients, we could replicate that on chips. Drug efficacy, therapeutic target discovery, new drug discovery, that drug, the TRPV4 inhibitors in phase 2 clinical trials, if not further. We've also done gene therapy delivery, adenoviral vectors on these chips. And it's won numerous awards. I am most proud that it won the International Design Award and beat out the Google car and a Frank Gehry building that year. But my family is most excited that it's now in the Museum of Modern Art's permanent design collection, which means I'm a permanent member of MoMA in New York City, and I get permanent discounts in the cafeteria and the gift shop. So now we've integrated immune cells. I showed you. We've also integrated stromal cells, like when we do pulmonary fibrosis models. And then we started to build other chips. So different pharma companies said, we're interested in asthma and COPD. Those are bigger challenges for us, and those are diseases of the airways. So we then made chips, this is Cambis Benham's work, where we took primary human bronchial epithelium on the chip, and we differentiated for 3 weeks under an air-liquid interface, and we later put endothelium on it as well. You get beautiful differentiation. Cilia move in a directional manner. If you put fluorescent microparticles to quantify mucociliary clearance, the bottom right, those particles are flowing in the mucus on chip at the same exact rate as in all of our lungs at this very moment. We then made chips with cells from patients with chronic obstructive pulmonary disease, COPD. And even after differentiating for 3 weeks, they retained the COPD phenotype, which is measured here by decreased toll R3 and 4 receptors. Now, what brings COPD patients to the emergency room are usually exacerbations, either by viral infection, bacterial infection, or cigarette smoke. And here we mimicked viral infection with poly-IC and bacterial infection with LPS endotoxin. And we could see that no response in healthy chips, but the COPD chips responded either to the LPS or to the poly-IC. And then Cambis, working with our engineers at the Wyss Institute, built a cigarette smoking robot. This is a Gatling gun of real cigarettes, a cigarette lighter from a car. It actually mimics true smoking parameters, meaning pauses, breaths, intensity, and timing. And now it delivers real smoke, not extract, right to the airspace. Now, this is just 2 simple examples, but on the left, you see IL-8, a major inflammatory cytokine. And I'll go over here because I can't see that, but on the left is normal. There's no change in response to cigarette smoke. On the right, you have a doubling of IL-8 in these COPD chips, which is very much like in patients. At the right, you see transcriptomic analysis, a gene microarray. The 3 lanes at the far right are from 3 different healthy donor chips after exposure to cigarette smoke, the changes in gene expression versus before. The 9 at the left are from a past clinical study of healthy people who also smoke cigarettes. We were really excited because we got pretty similar pattern in the top quarter versus bottom 3/4 of up and down expression. The reviewers were not impressed. They were very upset because ours were too similar to each other. We weren't mimicking the variation in patients and the intensities and so forth. And then we realized that we actually can do this type of study better than a clinical study because we were doing matched comparative modeling of the same patient before and after cigarette exposure, smoke exposure. So those changes are due to cigarette smoke, whereas the changes in these patients are due to cigarette smoke, but also work history, home environments, other things that they've done in their lives, and that eventually was accepted. And then we were actually asked to write a little commentary on how to do matched comparative modeling in vitro. Recently, Roberto Plebani was on sabbatical, and we got, with funding from Cystic Fibrosis Foundation, we were able to get cells from patients with cystic fibrosis. We put them on the chips, and at low mag, you could see there are more cilia here across the entire length of the channel. We also found that the beating frequency was increased versus normal, which is what's seen in patients, but you do not see this in a static transwell. You have to have flow. We saw increased mucus production as in patients, increased baseline inflammation in terms of immune cell recruitment. increased cytokine release. And then we looked at microbiome. We looked at Pseudomonas aeruginosa, which is in everyone's lungs, but it overgrows often in patients with cystic fibrosis, major cause of morbidity and mortality. And again, on the chip, we see much higher growth in these chips than in vivo, than in conventional systems. So one thing I want to emphasize, and I think so far today in the meetings I've been at, we're all focused on drug development, and that is basically how we all started in this field. I'm presenting our two-channel microfluidic model of MPS, but you're going to hear in this meeting there are so many different versions of this now. Each has its own strengths and weaknesses. But what we have found is that the real strength of organ chips and MPS is that they provide a window on molecular scale activities inside living human cells within a relevant tissue and organ context. And so this means that it actually can enable new mechanistic insights as well as drug discovery. And at the same time, you have an experimental system to test the predictions that you come up with that's human relevant. I think this is really the Intel inside is like the power of the whole of this whole technology. Now, a few examples. Brian Hassel was a grad student. I've worked on cancers since 1976. And big advance in that field was going from subcutaneously implanted tumors in mice to what it was called orthotopic cancer models, where you now take a mammary tumor and you inject it in the mammary fat pad, or a prostate tumor and you put it in the prostate gland, and it grows a little bit more like in human. So we had these human chips. So we said, let's make human orthotopic cancer models. So we took a human non-small cell lung cancer, cell line H1975 that a lot of pharmaceutical companies use. It's an adenocarcinoma form of this cancer that in patients emerges in the small airway, but it grows preferentially in the alveoli. And so we cultured it in the airway chips and the alveolus chips. We plated the cells at a low starting ratio to normal from the beginning. And what we found is quite interesting. On the right, the top is airway, the bottom is the alveolus. The gray dots show you the growth of these cells the way they've always been grown for 30, 50 years in plastic dish, serum-containing medium. They grow rapidly. The white circles is a plastic dish with the medium we use on our chips, which we only feed through the vascular channel because there's air above. There's no growth of these cells under those conditions. So all the growth I'm going to talk about is due to tissue-derived microenvironmental factors. Okay, so what we find on the airway chip is they don't grow at all for about 10 days, and then they start growing. And I will add that the airway chip is thick enough that we could go in with a needle and inject the tumor cells after the chip is formed, and there we get dormant tumors. But when we culture them from the beginning on the alveolus chip, they grow rampantly from the start. Now, what was really amazing is we did it with or without breathing motions, and breathing inhibited cancer growth on this chip by 50%. We could also measure invasion very nicely because you could just have GFP labeled tumor cells and watch them go through the pores, as you can see at the bottom. And we inhibited invasion by 50%. And using this system, we could show that the breathing motions regulate the therapeutic response of a first-line drug called ruxolitinib, a kinase inhibitor, such that when you treat the cancer in the alveolus chip without breathing, you have potent inhibition of growth. With breathing, it grows much more slowly, but it just keeps growing right at the same rate through the drug treatment. And we could show that this actually is regulated at the level of EGFR receptor phosphorylation. Now, we then started working with real viruses, and we were funded by NIH and DARPA. And the reason we were funded is that this is about 5 years, in 2017, there was enormous interest and concern about potential future pandemic respiratory viruses. And, but the focus was on influenza. So we developed a model of influenza infection. We took our airway chip. This is GFP influenza H1N1 at the right. You could watch the infection in real time. You could show that you get disruption of the epithelial junctions, cilia. You even get endothelial damage, which hasn't been studied before. We could use different viral strains, H1N1, H3N2, H5N1, and we could show that we get similar virulence as seen in patients. I don't have it here, but we've done it in COPD chips, which are 10 times more sensitive than healthy chips, which is just as seen in patients. We could quantify barrier function and show it's compromised. But more importantly, we could measure the host response. And this is essentially a cytokine storm that we hear about in COVID all the time. This is 6 different cytokines. The different strains of influenza induce cytokines to the same sort of levels that they do in patients. We could watch immune cell recruitment, as I showed you in the movie, but now we could watch them actually clearing virus from the channel above. And we could also measure drug effects. And in this case, we use Tamiflu. It's also known as oseltamivir, the first-line antiviral. And you have potent inhibition at a clinically relevant dose because we can flow drugs through at the clinical Cmax, not bathe the cells in it like in a static dish. We also did a really interesting experiment. Long C, who was a postdoc in my lab, basically mimicked human-to-human transmission of virus and showed that we could model spontaneous viral evolution of variants of concern, if you like. Basically infect the virus, in this case, influenza, and treat it with a drug at 90% efficacy. And here you see amantadine. And then after a couple of days, you take a droplet and wash it through the airspace and move it to another chip, just like you were coughing. And we do this again and again and again. And what we find with amantadine is that 8 passages, we get virus that's completely resistant to drug. When we do genome sequencing, we find all of the known mutations that have been published in patients who became resistant to amantadine clinically. And we identified about 3 mutations that no one's ever seen before. So that means you have the potential to see what's coming down the pike, which could have impact on vaccine development and therapeutic development. We did this with Tamiflu. It took 25 or 28 passages, but we got resistant virus, which is terrifying. We even got virus that was resistant to both, which is even more terrifying. Now, this is where we were. This is where we were. We started for the first time saying, could we use these chips to repurpose existing approved drugs for new applications so that you could get past the drug development 15-year timeline and do things quickly. And we went to the data that was published from Kambe's work in the smoking chips. And we noticed that there was many serine proteases that were upregulated. There was literature to suggest that they may be involved in the infection process. We tested 2 existing drugs, nifamostat, which is a anticoagulant, and trasolol. Both of them are broad-spectrum proteases. And we had potent inhibition of H1N1, H3N2 on the chips. And interestingly, we found that if we take one of these drugs, nifamostat, and combine it with oseltamivir, Tamiflu, we could double oseltamivir's therapeutic window from 2 days to 4 days. And this is important because FDA has only approved this drug to be administered in the first 2 days after you get influenza, which it usually takes more than that to diagnose that you have influenza. And so this actually could have clinical relevance. Now, this is where we were when COVID hit, January 2020. And Long C and Haiqing Bei are 2 postdocs who were trained as virologists in China and worked in my lab. They were following this on social media. The day after they published the gene sequence in January for SARS-CoV-2, they synthesized and designed and had synthesized engineered pseudovirus that expressed the SARS-CoV-2 spike protein so that we could use it in our BSL-2 lab. We don't have a BSL-3. They also quickly, and if you remember, those were desperate times, they went to the literature and they found 30 drugs that had shown some activity against SARS-CoV-1 and related viruses and Ebola. And they tested them, but they quickly tested them in an HUH7 cell line, which they had used when they were graduate students in virology, which is like the Vero6 line, is used because viruses grow well in them, likely because they have abnormal protective interferon responses. And out of those 30 drugs, as shown here, there were 8 that give you dose-dependent inhibition of viral infection in the low micromolar range, shown in gray bars. But what you might not be able to read is that 2 of these are chloroquine and hydroxychloroquine. This was February of 2020. Then we decided to test this in the organ chips. The first thing we did is we wanted to confirm that they express the ACE2 receptor and TMPRSS2, which is a serine protease that we now know is very important for SARS-CoV-2 infection. We had high levels of expression. Now we tested those same 8 drugs, and what we found is that chloroquine and hydroxychloroquine had no effect, nor did a number of others that eventually went to clinical trials. We found 3 compounds that did have some activity. The most potent was amodiaquine, which is another anti-malarial used for 50 years in Africa for prophylaxis. And we had funding from DARPA to expand this and collaborate with Ben Tenover at Mount Sinai and Matt Freeman at the University of Maryland, who have BSL-3 labs. And with Ben, we used a hamster SARS-CoV-2 model, and we could show that we get potent inhibition whether we give the drug prophylactically or after infection. And this drug, in part because of these data, is now in clinical trials across 20 sites in Africa. Separately, Hai-Ching in the group was working on the alveolus chip, and he was working on H3N2, and he did our classic experiment of breathing, non-breathing, and surprisingly, he found that breathing suppressed viral infection significantly. It suppressed cytokine production significantly. He did RNA-seq and many other studies that were published recently in Nature Communications, and he found that the one gene that was most highly expressed looking at breathing, non-breathing was S100A7. And this was important because there are S100 proteins that are related to this that were published, that were shown to scale with morbidity and mortality in COVID-19 patients as well. And it binds to a receptor called RAGE, it's receptor for advanced glycation end products, which is known to play a key role in inflammation. And we found a drug called aziliragon that's a RAGE inhibitor, and it potently inhibited the cytokine storm, basically, completely in our models. And this drug went through phase 3 clinical trials for Alzheimer's, oral, highly bioavailable, totally safe. They were like no side effects, but it missed its mark. And we reached out to the companies that have control of it. And this drug, the data shown here were included in an IND application to the FDA by a company called Cantex Pharma, who has control of this to start COVID-19 trials. So we're really hopeful that this will move along as well. Now, we've made many different chips. We started doing a human intestine chips in 2012, where we used CACO2 endothelial cells, an intestinal cell line that pharma's used for years. But I'm sorry, let me go back here. Yeah, now CACO2 epithelial cells, pharma uses them on trans walls, takes 3 weeks to get a poor barrier. They grow like flattened cells. We use the same medium that's been used for 50 years to culture them in trans walls and on 2D dishes where they have poor differentiation. And with peristaltic-like motions and trickling flow, we get spontaneous formation of intestinal villi in 5 to 7 days. Now we use organoids from from patient-derived organoids. We expand them in 3D gels. We then remove them, fragment them, place them on the chips, and we get absolutely beautiful intestinal villi. But now we can measure absorption, transport, mucus production, microbiome, et cetera. And this work was done by Magda Cassandra, who is here, and Alexandra Sontheimer Phelps and Youngjun Kim. Now, if we go back to the original studies with the CACO2, again, this originally came from a tumor. They grow rampantly in a poorly differentiated state on a plastic dish. Just giving them peristaltic-like motions and flow, they form villi. The proliferative cells move to the crypts, the bottom with EDU labeling. You wash out EDU, they migrate up to the top. You get all 4 lineages of small intestine. For the first time, they produce mucus. They have P450 activity. The only change was the physical, microfluidic, dynamic environment and peristaltic-like motions. We also found that, that bacterial overgrowth was regulated by the peristaltic-like motions. When I went to medical school, you're taught if the patient is anesthetized When they come out, you want to get them drinking quickly and eating so you don't get ileus. Ileus is bacterial overgrowth that leads to sepsis and death. Medical textbooks say it's because you're stopping flow. Well, we keep flow going. It's the peristaltic motions that stop, which you cannot separate in vivo. We can separate them on the chips. When we did, Magda did the primary organoid-derived chips, we compared the transcriptomic analysis of the organoid from the duodenum, the chip, which is the middle lane, and the in vivo duodenum, and we could show that the chip is closer to in vivo than the organoid from which it was derived. Alexandra made chips with colonic organoids, and this is a cross-section through the chip that shows you the morphology is amazingly reminiscent of what you see in vivo in goblet cells with magenta and apical microvilli. But now we could study mucus production, which is very important for ulcerative colitis, cancer, et cetera. And the cool thing is you could turn the chip on the side, visualize with differential interference, and you could see this white fuzzy stuff is mucus, and you could do this in living cultures over time. You just take it out every day, put it on its side, image it, and put it back in. And for the first time ever, we recreated the bilayer structure of human mucus and the thickness that is seen in vivo with a penetrable and impenetrable layer. Now, I think one of the biggest advantages of microfluidic systems is that we can culture living microbiome in direct contact with living human cells for extended times. You can do them in organoids for about 24 hours. You inject them with a needle and the lumen, and then they die. But we can do them for multiple days. And we published, Sasan Jalali Farizenhad was the first author on this work, where we created a hypoxia gradient by having oxygenated medium through the endothelial channel and nitrogen gas around it. And we can maintain oxygen levels below 0.5%, which means we can sustain obligate anaerobes in the lumen of the chip. We have oxygen sensors we integrated in the chip so we can measure this in real time. We've also integrated electrodes to measure tear in these chips. And so you have a gradient so that you could have obligate anaerobes in the middle, a mixture of obligate anaerobes and aerobes, and then you could keep the human cells alive as well. And with this, we published that we could take literally a stool specimen, collect a sample, dissolve the bugs out, put them on the chips, and we can maintain similar complexity as in human stool. This is over 200 different strains of bacteria from 11 different genera for 5 days on these chips. And the chips, if it's healthy from a healthy patient, the chips actually have a better barrier function. The right, it's hard for you to see, the little speckles are actually microbes. Now we have a paper that's in press at Nature Biomedical Engineering, where we're modeling environmental enteric dysfunction. This is malnutrition in low resource nations. And this has many factors, including malnutrition, as well as microbiome, as well as pathogens. And again, we can control these individually. And our initial results, we were able, with Gates Foundation funding, to get organoids from children with EED in Pakistan. They sent us the organoids and we put them on the chips compared to healthy. And the first thing we could see is that if you have healthy versus EED chips, we see decreased absorption of fluorescent fatty acids and proteins. What we do is we can culture these chips in complete medium or nutrient deficient medium, niacin and tryptophan deficient medium. And what we found, interestingly, at the right, this is transcriptomic analysis, we were lucky to have transcriptomes of the patients where we got the organoids from, and this, high and low different genes, which you obviously can't read. But what we compared were healthy versus EED chips in complete medium or nutrient deficient medium. And the net sum, as shown at the right, is only when you have the EED chip with nutritional deficiency do they begin to get more red at the top and blue at the bottom. And now we're beginning to integrate microbiome in these as well. And we actually have been able to model microbiome dependent effects. I don't have a picture here, but you get villus blunting, you get barrier disruption, you get increased inflammation on these chips as well. Another example about discovery is Alessio Travaglieri Larry is here at Roche now. When he was a grad student with me, we tested, we asked the question of why are humans more susceptible to enterohemorrhagic E. coli than mice? And you could have a small number of these bugs in a human and you get sick, and mouse can have orders of magnitude more and they don't notice it. And so we collaborated with Lacroix's lab in Switzerland, where they grew bioreactors with human versus mouse microbiome, complex microbiome, and they just gave us metabolites. And we tested them on our chips, and these are colon chips. The yellow are stained cells, and what you could see here is if you have the E. coli with human metabolites, you get desquamation of the epithelium, which is loss of yellow, whereas much less on the mouse chips. And we could then use this system with metabolomics to identify pretty quickly 4 specific metabolites that were responsible for this. Unfortunately, they were 4 in human microbiome that increased injury as opposed to 4 in mouse that we could add to human to protect. But it just shows you the discovery capabilities of these systems. So now there are other key goals that we've uncovered in the field, which I think remain for all of us. For example, we want to predict drug responses to drugs with clinically relevant doses. Right now, most publications on drugs in vitro bathe cells at high concentration for extended times, where in patients you have drugs go up and down, pharmacokinetic profiles, and there are clinical Cmax doses that our tissues see. That's not the way it's usually tested in vitro. We want to develop personalized disease models for individuals, create models that replicate complex immune responses, and also recapitulate species-specific effects. And so David Chu and my group developed a human bone marrow chip where we take CD34 cells from patients, bone marrow or blood, put them in a matrix gel in one channel with bone marrow-derived stromal cells, and then we put endothelium in the other and just perfuse through the vascular channel, and we get nearly normal neutrophil and erythroid differentiation over a month. This is a GEMSA stain at the right where you see multiple different cell types. We maintain the progenitor cells at much higher level than conventional cultures. We were funded by the FDA actually to do this, to look at radiation countermeasures and radiation toxicity. And here what you see is that we could replicate radiation-induced injury, but we do it at a clinically relevant dose, which is not what you see in animal models. Here you're in the 1 to 2 gray, and each dot is a different color. You probably can't see it, but these are 6 individual patient donor chips, and we get excellent reproducibility. We then used 5-fluorouracil, which is a cancer drug that causes marrow toxicity. Again, publications, marrow cells in culture, you see toxicity, but high dose, 5 days. In patients, they see it they perfuse patients IV 4 micromolar for 2 days. And when we do that on chips, as shown by this highlighted color, we see toxicity on the chips at that dose, but not in suspension culture or static cultures. And at this point, AstraZeneca came to us because they had observed a really peculiar regimen-specific toxicity in clinical trials. Very unusual for a pharma company to come to an academic when they have something in clinical trials. They had a cancer drug that, if given over 2 hours, you induce neutropenia and anemia. Same dose over 2 days, you only get neutropenia. Couldn't model it in animal models easily. They had the pharmacokinetics shown in gray at the top left, meaning they did mass spec to measure blood levels of the drug in their patients. And because we have flow, we could precisely mimic the PK in our chip, and we precisely mimic the difference in regimen-specific toxicity. Now, we use these chips to develop a personalized model with children at Boston Children's Hospital who have a rare genetic disorder called Shwachman-Diamond syndrome, where you have abnormal blood cell formation. And what you could see at the low mag is there's less cellularity in the chips, in these SDS chips. You could see quantifying less neutrophils, less red blood cells, less CD34 cells, and we saw abnormalities in neutrophil and erythroid maturity. But what was interesting is that we identified a phenotype never seen before, which was abnormal neutrophil maturation, low CD13 to C16 ratio. And this was never seen before. And so we went back to the patients. We only tested 2 patients, went back to 8 patients, analyzed their expression and found that there's 2 subpopulations in these 8. We happen to get 2 out of the lower ratio, but there's high and low ratio. We're not clear what this means, but we get basically insight into etiology of disease using these chips. Now, in COVID, this was the cover of New York Times Sunday section about a year ago, and it said there must be a better way because there was a shortage of monkeys to do vaccine testing. And it's like, wouldn't it be nice if you could just do this on human cells in chips? And so Garija Goyala in my lab did this. This was published recently in Advanced Science. We just simply take peripheral blood B and T cells, concentrate them in high concentrations, equal ratio in a matrigel in one channel of our chips, just perfuse medium through the other channel, and we get spontaneous aggregation of cells into clusters. And if we give an antigen, you get germinal center-like formation that has all of the appropriate cells. We literally took flu zone influenza vaccine that you could buy in a pharmacy, that we vaccinate the chips, we have dendritic cells there, and you get germinal centers, you get plasma cell formation, you get antibody class switching, you get specific IgG against the hemagglutinin antigen in the vaccine, and we could collect effluent and measure cytokines, and we were lucky that David Walt at the Wyss Institute happened to have clinical samples, and we could show very similar cytokine profiles in our chips versus humans. And we are now using this with GSK and other Gates Foundation and other companies to look at adjuvant testing, which can be modeled very nicely on these chips, as well as new vaccine development. Now, there were questions earlier about animals, and early on, when we were still at the Wyss, before we spun this out, Janssen Pharmaceutical and then AstraZeneca came to us and said, will you make rat, dog, and human liver chips? And we were really focused on no animals, human, but they explained that FDA often requires rodent and dog preclinical studies for drug-induced liver injury, and they often get conflicting results. And so we did this. This eventually moved to Emulate, the company we spun out, where most of the work was done by Kyunjin Zhang. But this was published a couple of years ago. And what you see here is one drug, bosantin, human, dog, and rat. These pharmaceutical companies knew the results already in animals, different species, and humans. And we precisely mimicked the toxicity in these 3 species. We also did multiple other drugs, and we were able to model cellular injury, steatosis, cholestasis, and fibrosis. But what I think is even more exciting, and this is largely done at Emulate, Lorna Ewert, who's here, and her team, is that Emulate has done, and they're now over 800 human liver chips. Each one of them has 4 cell types: primary human hepatocytes, liver sinusoidal endothelium, stellate cells, and Kupffer cells. And they tested 27 hepatotoxic and non-toxic drugs that were identified by the Pharma IQ MPS consortium that you heard mentioned earlier, which are pharmaceutical companies that have come together, and they published a paper that identified these 27 drugs. We know what they did in animals. We know what they did in humans. I think 17 or 18 had no toxicity in animals, but did in human. And if you could predict these better than animals, now we have something to talk about. So the results are pretty impressive. The chips predicted human drug-induced liver injury with a sensitivity of 87% and a specificity of 100%, which is about 7 to 8 times better than animal studies. And this has now been done in 3 different donors. I hope that this is the beginning of a change of how we do things and really reducing animal tests for a specific challenge. So as you heard at the Wyss Institute, we have, I think we're over 15 or 20 different organ chips between my group, Kit Parker, Chris Chen, Jennifer Lewis, and others. But many years ago, in 2012, I had a review that said, because we have endothelium line channels, we could collect them fluidically to create a human body on chips that would have a more physiological flow of compound across tissue-tissue interfaces in and out of tissues, which is really what PK is all about in the body. You can imagine an oral drug going to the lumen of the gut chip, watch it be absorbed, measure it be metabolized by the liver chip, peed out by the kidney. What does it do to the heart or bone or an aerosol drug in the lung? And we eventually did this with funding from DARPA. Richard Novak led the engineering on this. We don't connect by tubing because there's just too much dead space and it's too rigid. We use a robotic sampler, a liquid handler, and that could interact with the pharmaceutical industry as well. And you could go drop to drop from chip to chip. You could change the order. You could take one drop for mass spec to measure drug levels or cytokines, and then the next goes to the chip. And in this published study, we had, I think, 8 chips: gut, liver, kidney, heart, lung, bone marrow. Well, I can't even remember anymore what we had. We had brain neuronal networks from Kid Parker, and skin. And we kept them alive on this instrument for a month. But more importantly, the goal of the DARPA grant was to predict, based on in vitro data from chips in combination with computational modeling, human drug pharmacokinetic parameters, not correlate, quantitatively predict. That was the goal. It was a classic impossible DARPA goal. We did this by making a simplified human body on chips where we just made a first pass model of gut, liver, kidney, in this case, proximal tubule, and then we integrate an arteriovenous reservoir to mimic blood mixing when heart pumps. So we could take samples from the reservoir, and it would be like a peripheral blood sample rather than measuring a level after one particular chip or the other. And what you want to do here is to come up with parameters like the Cmax, the maximal peak drug concentration in blood or plasma, and the half-life. And we did this, and this was published. Anna Herland, who you heard speak earlier, led this work. We did 2 different drugs, oral nicotine and intravenous cisplatin. We had gut, liver, kidney, and this reservoir. Nicotine through the lumen of the gut chip, we measured with mass spec drug levels at all places. We collaborated with Andre Prequis at CFDRC, who's done physiological PK/PD modeling for pharma for years. So they had all of the blood flows and the masses of different organs. We just scale up from our little chips. And as on the right here, you can see that the dotted lines in color are predictions from the computational model. The dots with error bars are from a past study published in Sweden of nicotine gum versus 2 different forms of chewing tobacco called snus at 3 different doses. And we precisely predict the PK parameters there. And we did the same thing with IV with cisplatin with 2 different doses published in the past clinical study. So to summarize, we really believe that these chips that have 2 channels with an endothelium lining 1 faithfully recapitulate human pathophysiology, mimic human responses to drugs and radiation using clinically relevant exposures, quantitatively predict human drug PK parameters, and thus we think that they're really, we believe, ready to replace some animal models, both in the labs and in the drug development pipeline. And so, to end, I mean, these have many uses. They are this synthetic biology at the cell, tissue, and organ level. You could ask, is one cell type sufficient to mimic clinical response? If yes, you don't need all the rest. You want the simplest system that maintains all functionality that you're interested in. And this is what I call like one molecule of problem. Go further, and now you're down to atoms. But if you could do it with one cell type, God bless you. If not, something's missing. You add back endothelium, you add back immune cells, you add back microbiome, and you could control each parameter individually. I had a recent review of those interested in Nature Review Genetics. I think I have an 8-page table with 8 pages long summarizing, updating the field, and I know I missed a lot, I'm sure. But I ended with, the implications for personalized medicine. Right now, we have iPS cells and organoids that come from individual patients. We have primary cell lines. We can make single chips. We can make multi-organ human bodies on chips. We can model rare genetic disorders. But you can now imagine doing things like, toxicity testing, which would be, you can't do a clinical trial, for example, with lethal radiation, or it's very difficult to do trials with pregnant women. You could test male versus female. Clinical trials don't represent them equally. You could do different ethnic groups. You could do children versus old people. But you could also do what I think may be really the game changer. Right now, big pharma usually spends millions of dollars to do big clinical trials, and they almost always fail. And then they use statisticians to find out, is there like some subgroup, maybe a genetic subpopulation that responded better? And if so, they do a targeted study, and if they're lucky, they get approved for a narrow application. With organ chips, IPS, organoids, you can imagine flipping this on its head. Imagine you have a small group of, let's say, Hispanic women with COPD who are ultra sensitive to cigarette smoke. You get 50 of them, you make chips, you make their lung chips, their liver chips, their kidney chips. You find out which drugs work best for them in terms of efficacy, which are least toxic. And now you use those 50 patients for a targeted clinical trial. I think it would decrease the cost, shorten the time, increase the likelihood of success. And I think this is a different way of thinking about drug development. So that I must end, and I must disclose that as a Harvard faculty member, I have to disclose I'm a member of the board, chair the SAB, and hold equity in Emulate, which started in 2010 with our science paper and now has automated systems so that hopefully people get the same results wherever they are around the world. And I welcome you to visit their booth here. And thank you so much.